cells originated on Earth at a time when there was no oxygen in the ambient atmosphere, or almost none. And then after that, we have on Wednesday of next week the second quiz, and is due on Wednesday, and then the review session is on Wednesday, where we cover no new material, but you guys can have that be about going over textbook problems or reviewing material or whatever you guys want. So please come prepared to ask questions. And then the first midterm is Friday, and it's worth 74 points, 72 points out of a total of 400. So you should have been able to go in and look at your quizzes and figure out, and it should tell you which ones, what the right answers are and what your answer was. Is that accurate? Everybody can do that? Okay. So the scores were quite good on the quiz. The average in the class was somewhere a little above 14 out of 16. So the most common grade was 14 out of 18, 14 out of 18, so 7 out of 9 is the most common answer, most common score. There are some number of perfect scores, some 8s, and some 7 6s, and maybe a couple of 5s. So that was a good distribution, so let's keep it up. Yeah, any questions about any of the programmatic stuff, where we're going? Yes?
Is the quiz representative of what you think is important for the exam, or is it kind of just a...
It's like a snapshot. It's basically to make sure you're keeping up. Certainly the topics are representative. Online, so online there's a study guide now under, I think, this year's exams. It's not a comprehensive list of topics, so please don't look at that list and then come back to me after the exam and say, you asked me something that wasn't on that list. It's pretty close to comprehensive, but not, it probably doesn't have everything. Last year's exam is online. That's the best way to figure out what my exams are like. The only difference between last year's exam and this year's exam is this year's exam is shorter. Last year's exam was 100, this one's only 72. So I will not stretch it out to make it look, to condense 100 worth of points into 72. It'll be like 72 points, like the kinds of questions in the last exam. The answers of the last year's exam are also online. You might want to use the opportunity to practice an exam before looking at those answers. And the answers to the textbook problems of chapters 6 and 13 are also online. So, we're in the middle of chapter 13, and last time we talked about what I think of as one of the two most important principles of all of biochemistry, which is that cells have to keep their ATP levels far out of equilibrium with respect to the hydrolysis reaction that would form ADP and phosphate. In other words, that ATP reaction that forms ADP and phosphate, in the way we think of it, if we just think of it in terms of the chemistry, how much energy is in that high energy phosphate bond. We define that, as we talked about last term, in terms of the stability of the products that form after hydrolysis. So here are 3 reactions, not the ATP reactions, but 3 reactions that actually yield more free energy in the standard state than ATP hydrolysis does. And notice that, and this is phosphoenol pyruvate, phosphocreatine, 1,3-bisphosphoglycerate, first and the third are glycolytic reactions that we'll encounter on Friday. And these are all hydrolyses, right? So these are kind of like a comparative, it's like chemical comparisons of how much energy there is if you hydrolyze each of these high energy phosphate linkages. And the amount of energy that's in them, the delta G, right, this delta G is this with respect to the product. So really the high energy aspect of the bond is reflected in the stability of the products, which is why on this slide here, we commented as a high energy phosphate bond, which really means that the products are highly stabilized, meaning that they're low in energy so that the gap is big, right? So that you get a big delta G out of it. And if you get a big delta G out of it, then it's a high energy bond. And ATP has an intermediate level of energy that it comes out of hydrolyzing between the beta and gamma phosphates. Why not have ATP be the highest? Because ATP is serving as the cell's energy currency. And so if we want to make ATP, we need to have something that's higher than that in order to drive the unfavorable 30.5 kJ per mole that ATP synthesis costs in the standard state. Right? So there's got to be some reactions that are higher than that. Having said that, these sorts of reactions where you have, for example, a bisphosphorylated sugar that's a very high energy compound because of this acyl phosphate here, that is converted, that is used to make ATP in a coupled reaction, because this guy would yield -60 and this guy is only +30, so it would be favorable. Right? You make ATP on the surface of the enzyme that does that. That's called substrate level phosphorylation. And that is one way to make ATP in cells, but it only makes about 5% of the total ATP in cells. The other 95% gets made by a process called oxidative phosphorylation, which I will say something about in a few minutes. Okay. So any questions so far about sort of the structural basis, what high energy phosphate bonds, high energy bonds mean, why ATP has a high energy bond, its function as the energy currency of the cell. We could look at this. This is nothing more than a table of these reactions, which include the ones we've already looked at biogenetically. So here's ATP going to ADP plus phosphate minus 30.5 kilojoules per mole. This is just kcal column here. Some numbers are higher than that, some numbers are lower than that. So one thing to notice on this table, the sugar phosphates are low. These guys are above, as diagrammed on the other slide. But here, we've got acetyl-CoA, which is a thioester. And it releases as much free energy in the standard state when you hydrolyze it, as does the ATP hydrolysis reaction. In fact, very slightly more, minus 31.4. And it turns out that acetyl-CoA, as we will find out soon, is a very important cofactor in intermediary metabolism. And so we could look now at what is it about acetyl-CoA that makes this a high energy bond. This is why it's used. And the way to really understand it is to think of the hydrolysis as a comparison with oxygen esters. So if this is acetyl-CoA, and you notice here there's this CoA part, and I'm hiding the structure of it because it would take up this whole slide, this big structure, which we will encounter soon. But the business end of it here is just thioester bond. Right? So this S goes on to make the rest of them up. If you hydrolyze a thioester bond, you get CoA and then sulfhydryl, and then you get acetic acid. Right? So that's hydrolysis of this thioester, just like before we had hydrolysis of acyl phosphates or phosphodiesters, and we got free energies of hydrolysis out of that, and we classify things as high energy or not. And this one, when you do it, it does classify as high energy, minus 31.4. So acetic acid forms and hydrolyzes in this reaction. But this isn't really the reason why this is a high energy compound. So the way to understand it is to compare it to an oxygen ester. Okay? And the reason why the oxygen ester is not a high energy bond than the thioester is that the oxygen ester, as a substrate for the water, as a reactant with water that's going to be hydrolyzed, is itself stabilized because of this resonance. Right? So this oxygen in the oxygen ester can take on a partial negative charge, whereas the comparable sulfur in the thioester, sulfur is much less electronegative, doesn't take on a negative charge. Right? So this guy's up here, the oxygen ester is here. The hydrolysis in each case gives you pretty much the same products. This line indicates that the free energy of these products and these are pretty much the same. So this sulfhydryl and this oxygen, not much difference in the free energy of those two compounds. But the delta G is a lot bigger for this than for that. Right? So this kind of emphasizes that it's not only always about looking at the structure of the products. Sometimes if you're looking at these reactions and the molecule itself may have less energy in it than you might think because it has the capacity to be stabilized. Right? Before we talked about the stabilization of the resonance stabilization of the products. Now we're talking about the resonance stabilization of the reactions. Okay? So that's why acetyl-CoA as a thioester is a common cofactor in enzymatic reactions. right? And it gives us another metabolic role for sulfur. I mean, sulfur is a very important element. It's only, I think, the 6th most abundant element in cells, but besides cysteine and methionine, it kind of infiltrates itself in lots of different other places in metabolism. This is a key one, right, inside of acetyl-CoA. Okay, so on this slide, I just want to emphasize the mechanism by which ATP can make its high energy, make the energy in its phosphate bond available is very different in different metabolic contexts of the cell. You guys talked about myosin and actin last term? Okay, so you know there's ATP hydrolysis there, right? There's no chemistry reaction coupled to it, like synthesis of a sugar phosphate in that case. What we're using ATP hydrolysis for is actually the dragging, this physical motion of dragging these filaments, right, which requires physical force. Like there's resistance, there's drag in the cell. You got to slide something, you got to break a bunch of weak interactions and form new ones as you slide one filament past another, right? If you're talking about moving in a microtubule, it's transporting cargo inside of a cell, that's another example. So there's a number of examples. Sometimes also the conformational change in a large macromolecular particle has to undergo this big change. When you learn about protein synthesis next term, you will find out that GTP is used in the way, just as if it were ATP, and it's used in aspects of the whole translation mechanism that have nothing to do with actual peptide bond synthesis, but everything to do with the way the ribosome has to go through these complicated gymnastics to get from one conformation to another, which costs energy. So some of the energy of protein synthesis has to do with moving the ribosome around, so to speak, the parts of it with respect to each other. So that's one way, kind of what I said here. And then another way would be something like what we've talked about already, where we have hexokinase and we have glucose. Hexokinase is the enzyme. We have glucose, we have ATP. This kind of a direct reaction between glucose and ATP to form ADP and glucose 6-phosphate. We'll see this reaction again on Friday. Right? So that's a reaction where you don't really have an intermediate. There's no covalent intermediate. There's no intermediate in terms of the structures of the substrate. It's kind of just in and out attack and formation of products. Right? But if I showed you a reaction like this one, where we had glutamate going to glutamine, right? Most biochemists would be hard-pressed to understand exactly how ATP is used in this. Right? It's not a situation where you could say, oh, I have glucose, I have ATP, I know it's glucose 6-phosphate, the nucleophile must be the oxygen on the CH2OH at position 6. It's going to attack this phosphorus and ATP, right? You can kind of get a picture of what it is. But if it's this reaction here, right, you know, good luck figuring out what it is, because there's no obvious thing that's going on. Somehow this, you know, ammonia comes in here and substitutes for that oxygen. I don't know how that happens. Right? Unless we look here and we see how it happens. Right? So ATP is converted to ADP and phosphate, but it's not a hydrolysis where phosphate is formed. ADP is formed and the phosphate ends up attached to the gamma position of the side chain of glutamate. Right? So we get glutamyl phosphate. And glutamyl phosphate is an activated compound. Right? It's got this high energy linkage, and then ammonia can attack that carbon, and the phosphate can form, and this is its favorable leaving group, all that resonance, and you get glutamine. So it's just this two-step reaction. So sometimes the authors of the book are nice to you, and me, and write this out, and sometimes they're not. Right? In which case, we don't really know what the mechanism is, and you know, we have enough experience in biochemistry to take a decent guess that it would be something like this. But we wouldn't know unless we did experiments to try to isolate the intermediate, and really track the reaction and figure out what was going on. You should be able to do stuff like look at if glutamate reacts with ATP to form this compound, you should be able to look at the structures of these compounds and say, what is the nucleophile exactly? Like which atom is it? What's one of the oxygens on this like the terminal gamma carboxylated glutamate? What is the electrophilic center? right? The phosphorus, the beta, gamma phosphorus of ATP. Right? So you can name what those things are. Same for here, what is a nucleophile and what is an electrophilic center. Yeah?
So in the first step of the reaction with ammonia, where does the third hydrogen end up on the phosphate in the end? Because the product has NH2.
Probably. Yeah. Or it could get, depending on the pH and what else is around, it might end up on the enzyme, on an enzyme group. But if that happened, if we need to complete the cycle, it'd probably have to come back off the enzyme group. So yeah. So that's a, you know, you're pointing out an ambiguity around writing these reactions, which is that we don't always pay perfect attention to making sure that we really understand where where everything is coming and going from. And then you just kind of have to input some kind of common sense about enzyme reactions to think, well, it's going on a solvent, maybe it came from water, you know, maybe this PI got protonated, like you suggested. That's a perfectly good explanation. This is a bit of nomenclature. Just because ATP is so important, you hit these reactions all the time in biochemistry, so you need to know the names of them. This is showing an attack of this is 18O is a radioisotope of oxygen, which would be used in experiments to track the products. It's not really important for this slide. But you can imagine an attack of an oxygen nucleophile linked to something on the gamma or the beta or the alpha phosphorus atoms of ATP. And here you're forming ADP and transferring the phosphoryl group, the PO3. This PO3 part would be transferred to here, right? Here's PO3. This would be a phosphoryl transfer. Right? If it went here and you formed A and P instead, and you've got the whole pyrophosphate part, it would be a pyrophosphoryl transfer, obviously enough. And then the other common reaction is attack at the alpha position to release pyrophosphate and form this adenine ribose phosphate linked compound. Okay. And here we basically transferred all of this. And so this is called adenylyl transfer. Bit of a tongue twister there. So when we form pyrophosphate, not just in this reaction, any reaction that forms it, there's this special bonus in cells, because almost all cells have this enzyme called pyrophosphatase, and that takes PPI and cleaves it and hydrolyzes it, binding water, binding pyrophosphate, forming 2 molecules of inorganic phosphate. So this PPI goes to 2PI by pyrophosphatase. This enzyme's almost always around. So if you form PPI in a reaction and then pyrophosphatase immediately removes it to PI, then by mass action, you can help drive the formation of more of this. Okay? So it's a way of immediately helping to drive a reaction if you form PPI. And this is very important in, for example, nucleotide synthesis. This is the synthesis, in this case, of RNA via template-driven reaction, where we're adding this G and pairing to a C on the other strand, for example, or whatever the other strand is. And so the 3'OH attacks the alpha phosphorus of GTP in this case, right, and forms the phosphodiester linkage to lengthen the RNA chain, and it produces pyrophosphate, and then the second anhydride bond is broken here to generate 2 molecules of inorganic phosphate. Right. So this lowers the pyrophosphate concentration, so it's going to improve delta G because it's going to improve that Q term. Remember, from delta G equals delta G0 plus RT ln Q. And Q is the concentrations that are actually in the cell. If the PPI concentration is low because pyrophosphatase eats up whatever PPI forms, then that's going to help the reaction drive in the direction of forming more pyrophosphate. Right? So this is a big driver for synthesis of nucleic acids in cells. Questions? Okay, let's move on. All right, so the last sort of topic in the sort of conjunction of the whole collection of the things that we talked about here in the beginning of the class is something about redox reactions. It turns out that you know, for all the attention that we're going to give right in the beginning, we're going to give a lot of attention in glycolysis. This starting with glucose, and we're going to track through this pathway, and we're going to, you know, we're going to learn all the structures of the compounds in the pathway, and see where the ATP is made on the enzyme, and think about which reactions are favorable and which ones are not, which ones are regulated and which ones are not. For all of that attention that we give to the glucose pathway itself, along the way there will also be some redox reactions. And the redox reactions have the effect of moving electrons from fuel-rich molecules like glucose. And later in the term we'll learn that it's the same thing for fats. These are fuel-rich molecules that are effectively burned in the cell, right? And so electrons are removed from them, okay? And so they're oxidized, right? And they're transferred to some acceptor, which of course is then reduced, so you have a redox reaction, right? So a lot of the redox reactions in cells have to be understood in terms of looking at the structure of a carbon substrate as you go through the glycolytic pathway, for example, and other pathways. So that we want to be able to recognize when we look at a carbon-containing molecule that's mostly uncharged, or if it is charged, it's not really relevant to this redox thing we're talking about, to see what is that an oxidized, how oxidized is that carbon-containing molecule, that sugar in the middle of the glycolytic pathway, as compared to some other carbon-containing compounds. And so this table is kind of a bit of a cheat sheet to tell you about that if you're not familiar. You know, there's no charges here. We're used to thinking of redox, perhaps, in terms of you know, what you learned in freshman chemistry: iron 2, iron 3, oxidize it, remove an electron, go from iron 2 to iron 3. You know, the whole electron's been removed, so it's obvious what the redox state has changed. You've got a number on it that tells you it went from +2 to +3. So there's no numbers like that because there's no actual formal charge on these carbons. Nonetheless, we're saying that this carbon, this red carbon in ethane, is more reduced than this red carbon in ethanol. So that carbon got oxidized in the process of going from ethane to ethanol. And this gets associated with this number 7, which basically says if you've got a carbon bound to a hydrogen, we'll count both electrons as if they were part of the carbon. If the carbon is bound to another carbon, we'll count one of them, and the other carbon gets the other one. But if the carbon is bound to an oxygen, it doesn't get either one because the oxygen is more like electronegative. So this is, you know, this is pretty quick and dirty here. It's not really very accurate, but it provides a mnemonic or just a tool to say, well, you know, here there's only 5 little red dots around the carbon, and the number's lower, so lower numbers mean more oxidized. In this scheme of counting the electrons that are on the carbon, because they've basically been taken away. Right? So oxidation, removal of electrons. Right? So this carbon is oxidized. Usually, when the carbon is oxidized, you can recognize it because it's bonded to something electronegative, often oxygen, sometimes nitrogen. Right? So alcohols are more reduced than aldehydes and ketones, and acids are more oxidized yet. This is a little bit of a misnomer here, because usually we think of a carboxylic acid as being more oxidized than the ketone. So I think this table breaks down a bit there. Here you've got a carboxylic and acetic acid where the number is 1. Here you've got a formic acid and the number is 2. So this is only showing the limitations of this way of diagramming things. And then the most oxidized form of carbon, of course, is CO2. There's still no charge on the carbon compared to, say, methane, but we consider this to be super highly reduced and this to be super highly oxidized, even though there's no charge on either one. It's just a little delta plus or delta minus.
So it's the same with nitrogen, basically counted the same as oxygen.
Yeah.
Nitrogen takes all.
Yeah. So the other thing we can notice here is that oxidation occurs together with taking away hydrogens. So dehydrogenation means remove hydrogens. And you can see that here, for example, the hydrogen to carbon ratio in methane is 4. The hydrogen to carbon ratio in carbon dioxide is 0. And as you go down this table, the hydrogen to carbon ratio goes down. So as you get more oxidized, you have less hydrogen hanging around. So that speaks to one of the key mechanisms by which the electrons are actually removed. Okay. Any questions about this? Okay. Ways to transfer electrons. So if we're talking about redox reactions, we're talking about the transfer of electrons back and forth between compounds. There always has to be a donor and an acceptor. So how could we transfer them? These are the 4 ways that we can see that they're transferred in biological systems. So one is as a hydride ion. There's a pair of electrons here. Okay, and there are reactions that we will look at where there's hydride transfers, and that is a way of moving electrons around. We could imagine that they could be transferred directly as electrons. So there are going to be chains of electron transport that we will study that involve proteins, but on those proteins there'll be metal centers. And so the electrons can get moved between metal centers and oxidize and reduce the metals in the metal centers on the proteins. So that's another way to move electrons. Okay. We can move them as hydrogen atoms. Okay. So here we've got the donor molecule, which we're just symbolizing as AH2. And we're stripping away electrons and protons at the same time. Right? So this is perhaps the most common mechanism. This would be what we would think of as a 2-electron transfer. Right? We've got AH2, we're taking out 2 electrons, and we're taking out 2 protons. Right? And so this is also a dehydrogenation reaction because we're taking out the hydrogen. So this is, this is just written very schematically, but we will see specific examples of what this means. Right? And then the last way is to actually directly combine with oxygen. So I think you can still see this. Here's an electron donor. Oxygen is the best electron acceptor in the cell. So when we burn fuel molecules with oxygen, we're moving electrons to the molecule that wants them more than anything else, more than any other molecule wants them that's around in the cell. Okay. So if we transfer directly to oxygen, and there are different kinds of enzyme classes that do this, and we'll encounter them later. There are oxidases, and then there are oxygenases. We don't need to worry about the distinction now, but it depends on some details of what the substrates and products look like. So here we've got a methyl combined with molecular oxygen, and we've generated an alcohol. Right? And so the oxygen has become reduced. Here it's the most oxidized, it's become reduced. The carbon has become oxidized because it's now bound to an oxygen. So this is a redox reaction, basically. It doesn't look like a conventional kind of redox reaction, but it's certainly a redox reaction for purposes of how we think about biochemical metabolism. Yeah?
And are these reactions balanced the same way as we learn in general chemistry?
Yeah, generally you can balance them the same way. We're not going to be balancing them. But yeah, you add protons and electrons and waters to the 2 sides in a defined order. Yeah. But we don't need to go there for purposes of this class. And I think you'll probably find, as you look through the book, that the textbook authors are not particularly rigorous about balancing them either. So here's our standard reduction potential table. Okay, so this is exactly like the table you would have learned about in freshman chemistry, except that it now includes a whole bunch of stuff that wasn't in the table before, but nonetheless still functions as redox reactions, right? So So here's the standard reduction potential, E prime 0V, for a given half reaction, which is a measure of how much it wants to take electrons. So you notice that they're all written as reductions. This whole table is written as reductions. It's a reduction potential table. And yet, of course, when we're talking about a redox reaction, there's always a pair, right? If something's getting reduced and something else is getting oxidized, and then we want to predict what's the direction of spontaneous transfer. And when we do that, right, if we have one reaction up here, one reaction up here, the equation that we need to remember is delta G is minus NF delta E. So really this table of reduction potentials is exactly the same in principle as the thermodynamics of phosphoryl transfer, where we have delta G's going from ATP to ADP plus phosphate, or any other reaction. We have it's all thermodynamics in both cases. We have one molecule, we have molecules of substrate here, we have molecules of products here. There's a difference in delta G. Right? The only difference here is that we move from one to the other by transferring electrons, as opposed to some more a reaction that the enzyme catalyzes, which of course also involves a redistribution of electron density in the molecules. It's just that here we think of it as a direct movement of the electrons. But it's still thermodynamics. We can measure it in terms of volts, but we convert the volts to delta G right away. Right? So this is nothing more than like your ATP free energy is minus 30, your phosphotriachin is minus 50. Here this reaction between this pair and this one is however it will come out based on the math of this table, which we'll look at an example of. Right? And the math of this table will give you a delta E, and then it's converted by the number of electrons, Faraday's constant, which is a conversion factor. Yeah, that gives you the free energy. Okay. So I'm sure that most of you, or all of you, have seen this table. Right? But I think it's good to recognize that when we have a table like this, it's a thermodynamics table, not a kinetics table, and it's the same kind of information as the delta G's that we're talking about just earlier today when we were writing them out for these phosphoryl transfer reactions and any other reaction you see. That delta G and this delta E are the same kind of thing, right? So, if we're looking at bioenergetics in terms of what's going on with in glycolysis, this is what's going on in glycolysis. Actually, more than just glycolysis. This is what's going on in aerobic respiration in cells that live in oxygen-rich environments. They're burning their fuel, right? So let's say that their fuel is pure glucose, which you could dump into your car engine. It wouldn't work very well. It's not built for glucose, but there are engines that run on sugar as fuel instead of oil as fuel. And then, of course, we could write an equation like this for oil, which we will do because we're studying lipids as well. But let's write it for glucose, because that's going to be where we start with glycolysis. And we'll learn that glycolysis extracts this tiny little fraction, it's like minuscule little bitty bit, like 5% or something, of the total possible energy in the glucose molecule gets pulled out in glycolysis. Right, so this is the complete oxidation of glucose. Right, so this is a reduced energy-rich compound. Right. This is a highly oxidized compound. Here, this carbon has become oxidized. And the water is the reduced form of the oxygen, having also picked up a bunch of protons. So this is reduced. So this is thermodynamically downhill, and that's putting it mildly. Right? So obviously, huge amounts of energy that propel cellular life, at least heterotrophic life, by burning the glucose in the cell. And so this is going to be, energy is going to be extracted in a internal combustion engine burning oil or sugar or whatever it's burning. This reaction occurs all at once at conditions of high temperature and pressure. And a lot of the internal energy of the glucose molecule is wasted as heat. To the extent that we're smart engineers, we can couple that energy release to the movement of the car or whatever, but we're still wasting a lot of it. Whereas the cell wastes a lot less of it. And further, the energy is extracted in small pieces. So if the total amount of energy in there is something like 3,000 kilojoules per mole, and we want to extract the energy in pieces, what would be a good sized piece that you would want to extract the energy in? Like what number?
500.
500, you can only get 6 pieces if you got a total of 3,000. So that's a pretty big piece. You're probably going to waste a huge amount if you get a piece as big as 500. What number are we familiar with that would be a good piece that would correspond to what the cell's actually doing?
30.
30. So how about a piece that's, say, 30.5 kilojoules per mole? How about a piece that's exactly that big? Right, because of course then we can do this. And so this will happen, as I mentioned, by substrate level phosphorylation, right, which we will see examples of on Friday. Or it can happen, so in a substrate level phosphorylation, for example, there would be a compound like 1,3-bisphosphoglycerate going to 3-phosphoglycerate, where here there's 2 phosphates, here there's 1, and the other one ended up on ATP. And this hydrolysis here is highly favorable. And this synthesis is unfavorable, but not by as much. And so this is a couple reaction by which you can actually make ATP on the surface of this enzyme, which is one of the later steps in glycolysis. Right? So as I said, this kind of thing is getting you maybe 5% of the total energy in the cell. And the other 95% is done by this process of oxidative phosphorylation. And the way oxidative phosphorylation works is that there's been a transfer, is that these energy-rich fuels like glucose transfer electrons to redox cofactors like nicotinamide adenine dinucleotide, which we will see the structure in a second, and then they reduce that cofactor. And then that cofactor interacts with something else and reduces that, which reduces something else, which reduces something else. So the electron's getting passed down and down and down and down. And ultimately, if you're in an aerobic cell, the very last electron acceptor is oxygen. So there's an electron transport chain, which starts with the reactions that we're going to learn on Friday. And the key aspect of the reaction, one of the reactions we'll learn on Friday, is that we're going to synthesize this molecule called NADH, which is the reduced form of this cofactor. And that NADH, being reduced, can reduce something else and become oxidized, and then go back to glycolysis and get another electron, actually a pair of electrons. Right? So oxidative phosphorylation works by an electron transport chain which delivers electrons through redox reactions to the mitochondria, at least in mammalian cells or animal cells, cells that have mitochondria. And then the energy in these transfers is used. Sorry. So, electron transport chain. Electrons to mitochondria. Build a pH gradient across the inner mitochondrial membrane. Use the pH gradient to drive ADP plus PI goes to ATP. Okay? So that's the process of oxidative phosphorylation. Maybe you got it in outline if you took a cell biology course or something like that. We're going to look at that process in some substantial detail. Okay. And this is on the order of 95% of the ATP in an aerobic cell will be made this way. Okay. So this is kind of like preliminary, so you just kind of get a sense of where we're going and you have some more context for what we're about to do. Yeah.
To like keep homeostasis, right, at a certain temperature, is this related to like the heat that gets released along the way?
There are mechanisms that conserve heat in the process, especially in newborn infants. There are decoupling processes associated with electron transfer that deliberately, apparently, waste heat so that newborns can stay warm. That's pretty neat. But in general, the cell doesn't waste a lot of heat. I mean, to the extent that things warm up, you've lost some of the internal energy of glucose, and you haven't done anything except increase entropy. So, yeah, generally you don't want that. Okay, so we're going to unpack this throughout the rest of the term, right? But this gives you a sense of kind of where we're going. So let's look at some structures for these cofactors. So this is NAD. I don't expect you to be able to, you don't have to be able to draw it from scratch. You have to be able to recognize it and understand how it works. And we will see some aspect of the mechanism soon. But the structure of NAD, this is the oxidized form. And notice that it's got an adenine ring, a ribose type sugar, right? Whenever we find these cofactors like ATP and NAD, and they always have sugars in them, the sugars are always ribose, kind of like reflecting the RNA origin of life kind of deal. At least that's one of the pieces of evidence that gets pointed to. There are two kinds of nicotinamide cofactors, NAD and there's NADP. And the difference between them has nothing to do with the chemistry of the redox part. It's just that NADP has this extra phosphate group, which helps basically bind it to the enzyme. Okay. However, having said that, it's also true that when we look at the picture of metabolism more broadly, NAD will get used in certain metabolic contexts, and NADP will be used in other ones. And so the cell is very careful about keeping them apart. Even though as far as their redox function goes, it's the same. Right? So that phosphate is down here when it's present. And then it would be just NADP plus. Right? The plus here, of course, you've got negative charges on there, so you could say, well, I see a plus there, but two negatives, so it should be NAD minus. But by convention, that plus refers to what's going on in the nicotinamide ring only. So we kind of forget this. This is like so much cabbage going on out there. Although, of course, it's important for any enzyme that binds it, it's going to have binding determinants on the cabbage. So this is the oxidized form of the nicotinamide ring. So you can see what the structure of that ring looks like. It's a heterocycle with one nitrogen in it. Right? It's got this amide coming off one position of the ring. And it accepts 2 electrons and 2 protons to form an uncharged structure that also has an additional hydrogen on here. And a proton is released in that mechanism. So we'll see how that works in the context of a particular enzyme, which is a dehydrogenase. So the class of enzymes called dehydrogenases are enzymes that carry out redox reactions, i.e. we had 6 categories of reactions, or 6 categories of redox. Oxidation reduction was one of those categories. Dehydrogenases are a common enzyme in that redox category, and they use NAD or NADP. Right? And they convert it to NADH. Once they've converted it, you should ask, this is a generalized reaction here. Where these protons could come from another substrate, they could get picked up from solvent, for example. Right? Depends on the mechanism. Where are these electrons coming from? Right? So something is delivering electrons to this. Something else that is reduced is delivering electrons to this. Right? And in the context of glycolysis, of course, what's reduced is the sugar. Right? So it's not going to be glucose directly, but somewhere along the line, electrons are coming out of the sugars and getting into NAD plus to form NADH, which is then going to go on and do its thing in the mitochondria. Right. This is just showing you that there's a different absorbance spectrum for NAD plus versus NADH, and that means that it's easy to detect them in enzyme reactions. So if you got a dehydrogenase and you want to assay it and you have the right substrates, add the enzyme, close your spec, hit return. The data accumulates in your computer as the reaction goes on. Okay? If you're forming NADH, the absorbance at 340 is increasing, and all you have to do is track it, and next thing you know, you've got kCal. So these are the important facts about NAD that you should know. It's soluble and diffusible in cells. That's very, very important because some cofactors aren't, and that controls or shapes how metabolism is set up. Okay. The substrate is oxidized. The substrate meaning whatever the other compound is in the reaction. So NAD is, if you can think of it as a substrate, you can also think of it as a cofactor or both. It doesn't much matter if you call it a substrate or you call it a cofactor. But there will be some other molecule in there, like some sugar compound. And that sugar compound will be getting oxidized according to the rules we learned on the first slide, right? So that this guy can get reduced. And that'll be one of the steps along some metabolic pathway, right? So the substrate is oxidized, and hydrogen atoms are also removed from it. If not two, then maybe only one, and one hydrogen comes from solvent, something like that. But often 2 hydrogens are removed from substrate, and 2 electrons are removed from substrate. 2 electrons and 1 proton are transferred to NAD, the oxidized form, to reduce it. And the way that they're transferred is as a hydride ion. So remember, that was one of the ways in which we could transfer electrons. Okay. The remaining proton is released to solvent. There can also be, in some cases, some stereospecificity of hydride attachment to the ring. So this ring, if you look at it from the front or you look at it from the back, it's a little subtle. And this is not something that I really want to dwell on, but the textbook goes into it a bit. Sometimes these dehydrogenases will, in the details of the mechanism, come from one face of this ring or come from the other in terms of adding electrons to it and adding proton to it. Okay, so those are the important facts about NAD. And I mentioned, or most of them, I mentioned that NAD and NADP have different roles in cells. In oxidations, like what we're about to learn for glycolysis, it's NAD that's more used. In anabolic reductions, meaning biosyntheses, right? Anabolism is biosynthesis. Reduction means you're making something, you're reducing it, right? Those reactions tend to use an NADPH. So we'll see examples of that. Burning fuels and synthesizing new fuels, right? Or synthesizing large components to incorporate into the cell, if they're not going to be fuels, are separately compartmentalized in cells. Like burning fuels is generally compartmentalized in the mitochondria for aerobic cells that have mitochondria. I mentioned that the electron transfer reactions and the synthesis of ATP go on in the mitochondrial membrane, so it's efficient for the processing of the fuel molecule to also go on in the mitochondria. Okay, yeah?
I was wondering, what is the effect of releasing that extra proton inside the cytosol? Is there a way to regulate the pH so it doesn't?
Yeah, there are. Or it would get picked up by something else. Everything's buffered, so you don't have to worry about the cytosol getting too acidic because of that reaction. Okay, so that's NAD. NADP. Here's a reaction as an example of a coupled NAD-dependent reaction. Pyruvate, as we will see on Friday, is the very last metabolite in the glycolytic pathway. Like after step 10, there's 10 steps, or 11 steps if you consider this one as well. Right? You can think of this as the 11th step of the pathway we're going to learn on Friday. But I'm just showing it now because I'm just using it as an example. Pyruvate is the more oxidized compound. It emerges from 10 steps of glycolysis. This is what it looks like. The enzyme that does this reaction is lactate dehydrogenase. You might remember lactate dehydrogenase because it's the example we used to talk about isozymes, A4 and B4, back in the first week somewhere. And so NAD is used here. It's not written on the slide. But to go from lactate to pyruvate, you remove 2 protons and 2 electrons. To go from pyruvate to lactate, you add 2 protons and 2 electrons. Okay. So for reasons I will talk about on Monday, this is called a fermentation reaction. But right now, we're just looking at it as an example of an NAD-dependent reaction. And so we can also use it as an example of how to use that table, right? Which is so easy and hard to feel like I should spend the time to do it, but I will anyway. So this table is the conventional table, and we could say, if we go to that table and we look at it, we will see that we have this reaction on it. And we also have this reaction on it. Right? And in order to decide what is spontaneous, we always have to have 2 reactions going on because we have to have an electron donor and an electron acceptor. Redox means we can never just have 1/2, right? But we're going to pull electrons away, but they don't go anywhere, right? Makes no sense. Right? So there always has to be 2 reactions. And we want the delta E, we want the difference to be positive because we know that delta G is minus NF delta E. So if this is positive, right, then this is going to be negative, and then that'll be spontaneous. So this is all in the standard state, right? But we will be able to predict the direction in the standard state, which is what you do, everything we've done so far in terms of predicting 3 reactions. Starts with the standard state at least. So this would turn into pyruvate plus NADH because we're going to flip this one. Right? Pyruvate plus NADH goes to lactate plus NAD plus 2 electrons. And so then these electrons cancel out. And so delta E is plus 0.135 volts. Okay. So just like there's an equation that says delta G equals delta G0 prime plus RT ln Q, here there's an equation that says E equals E prime 0 plus, takes a different form because we have to work in Faraday's constant. So this is the Nernst equation. And this is the equation that you would use if you're not at standard state and you're dealing with electron transfer reactions. Okay. And so F, the Faraday constant, which of course, if you need to give you on the exam, is N is the number of electrons transferred. Which can be a way to screw up a problem if you don't pay close attention to what's actually going on in the reaction. Right? Don't just assume N is always 1. Okay, so that's all that we need to talk about in terms of this. So the richness of this is how this redox fits into the metabolic scheme, not about the details of this computation. It will not be any more complicated than this. I mean, we could notice in this table that we've got this standard state value. And here we've got Fe3 plus going to Fe2 plus. We've got it here. We've got it 4 places, and the value's different in these different places, even though it's Fe3 plus Fe2 plus all the time. And then, isn't there one more over here somewhere? Yeah, right here. So, what's up with that? If this was like, freshman chemistry table, and it's Fe2 plus Fe3 plus, there would be one number, and that would be it. So how can there be like 6 different numbers for what is apparently the same redox reaction? And here it's all cytochrome. We'll learn what cytochromes are soon. Yeah?
Because of the Q factor?
No, this is all standard state. Any ideas?
Different enzymes?
Yeah, different protein environment, different different capacity to take or give away electrons depending on the electronic environment of that particular cytochrome and the proteins around it, the protein structure around it. Right? Yeah. So think of pKa shifts. Right? pKa of aspartate shifts in the active site because the electronic environment is different. The reduction potential of a particular redox cofactor, the extent to which it desires to grab an electron, right, is different depending on what's around it, right? Just like the desire of aspartate to grab a proton could be different in an enzyme active site than it was just in solution, leading to the pKa shift. Okay, so that's an interesting thing to look at. It gives you a little bit of further context in terms of that this is be a little more complex than just looking at the numbers. So finally, I want to show you, this is one more cofactor. We're going to look at more of them, but this is enough for now. So this cofactor is called FAD. And this portion of it here is called FMN. FAD is flavin adenine dinucleotide. So again, we've got this adenine nucleotide portion of it. Ribose again. It's linked through some aliphatic chain. And then at the end, we have what's called this isoalloxazine ring, which is the business end of it. Right? So sometimes the book may talk about FMN or FAD, but as far as the business end goes, it's just, for some reason, whatever the context is, it's decided to refer to this instead of that or the other way around. Usually it's FAD we talk about. And so the couple properties of this here. One is that this is called a flavin. There are other kinds of flavins, by the way. The only kind that we need to worry about for this class is FAD. But there are types of flavin-like molecules, FAD-like molecules that also do redox reactions in other kinds of cells, like anaerobic cells. But here you notice that there's a single proton and a single electron transfer, and it forms this sort of intermediate structure here with a single free electron, right, which is not a stable structure. Typically, this would be highly reactive. It would be like a free radical structure. Right? So an unpaired electron here is called a semiquinone intermediate. And then this can take another proton and electron and end up with a fully reduced one. So usually we're talking about FAD and FADH2, but there are going to be contexts in which we have to worry very directly about, is it one electron transfer or two? Right? So sometimes electron transfer reactions go with pairs of electrons, like NAD, which is a pair of electrons from pyruvate to lactate. Right? But in other contexts, it'll be just one electron transfer. And sometimes you need to go from a situation where you have two to a situation where you have ones, in which case you need something that can do both, two and one. Like it's going to accept 2 and give 1 away, and then give another 1 away later. Right? So that's the kind of thing that FAD is good for. Right? So that's one big difference between FAD and NAD. And the other very big difference is that it's very tightly bound. Right? So in this case, we don't think of FAD as a substrate, typically. Sometimes I might get lax and say the substrate is FAD. But the reason that it's probably not good to think about it as a substrate is that it's not diffusible. So we said NAD was diffusible, which is, everything is diffusible, right? I mean, glucose is diffusible. All the substrates and products in the pathway are diffusible in the cell, typically, right? But sometimes there are exceptions, and this is a biggie, right? So it doesn't release, right? If you go from FAD to FADH2 and you reduce FAD, now you have FADH2 and then you want to do something with these electrons, like get ATP out of them somehow, right? You're going to have to bring the protein molecule with the FADH2 on it to where it needs to go, right? Or the cell's going to have to localize it there to begin with, because the FADH2 is not going to dissociate and drift over to where ATP gets made or it's needed for something else. It's stuck on there. So it just cycles between FAD and FADH2 on some enzyme. And depending on the enzyme, the E'0V is going to be different. Just like the E'0V of Fe3 plus Fe2 plus was different in that table, 6 different ways. I could show you 6 different FAD enzymes, and the E'0V would be different. There's one number in the table. Right, you can find it in here. Here. We don't have any other ones, but this. Notice that there's a little star here. Star. This is the value for free FAD. FAD bound to a specific flavoprotein, right? Some enzyme in the cell that uses FAD, but also binds other substrates, right? Like succinate dehydrogenase, an important enzyme that we will study, has a different E'0V that depends on its protein environment, right? So this little asterisk here, turns out to be very important for understanding, for cluing us in about how FAD works, different from how NAD works. Okay. Of course, there are pathways in cells to make FAD and NAD. Right? We don't talk about them in this class. They are what we could think of as something called secondary metabolism. We're only talking about primary metabolism. We're talking about like the big guns here, like glycolysis, TCA cycle, some aspects of nitrogen, like urea cycle, like the big names. Right? But underneath that, you have all your hard-working army of enzymes that are doing things like providing NAD and FAD so that the big name pathways have them to use. Right? FAD is a big complex, it's a huge molecule. I mean, imagine what the pathway is like to make it in cells, what the starting material is. What's the starting material going to be, like bicarbonate and ammonia or something? Right? I mean, what's the cell growing on? Glucose? There's nothing that looks like glucose in here. I mean, this is going to be a huge pathway to make this thing. Right? Costing a lot of energy. So this can help to keep in mind the fact that we've got a lot of this stuff is going on in the background, and while we're not talking about it, we know it must be there. Questions? Yeah.
Maybe if FAD, once it's made, it's generally a status, just keep producing it and recycling it.
Yeah. Yeah. It stays bound to an enzyme, and it probably has the same lifetime as the enzyme itself does. Yeah.
You called the semiquinone an intermediate. Would you call FADH2 in its fully reduced form also call that an intermediate?
I don't know if I want to call it semiquinone intermediate. Did I say that?
You did.
Okay. No. Why don't we back off on that?
Okay.
I like to think of an intermediate as something like an isolated study, and I'm not sure. While we know that that exists and it goes through that, I'm not sure how isolable it is. So we could say it's a short-lived intermediate. It's better than that intermediate they talked about in the chimal mechanism. That's definitely not an intermediate. Okay, very good. So we're caught up, and we will get into glycolysis on Friday.